Hi, my name is Jenny Rohn, and I'm a cell biologist at University College London. Bacteria have been on the planet for billions of years longer than we have. Because of this, despite their tiny size, they're very good at infecting us. And I'm interested in how bad bacteria can trick us and make us sick. So one area where bacteria have the upper hand is in patients who have received a donor kidney. Up to 2/3 of all kidney transplant recipients will experience a urinary tract infection, or UTI for short. This can cause loss of the donated kidney, sepsis, and even death. So as such, it has a huge healthcare economic cost and human cost. So the problem of antimicrobial resistance is actually making it worse for kidney transplant patients. So what is AMR, or antimicrobial resistance? So when bacteria are dividing like these, they can be killed by antibiotics, zap, but bacteria can actually express a gene that prevents this from happening. It's like a defense shield, and that's what antimicrobial resistance is. So in the donor kidney environment, you can see here that the kidney is attached to the bladder, so the urine can come out. But you have to keep in mind the environment in which this happens, and that's in very close proximity to the gut. So the opening to the gut is very close to the opening to the bladder. Within the gut, of course, there's lots of bacteria. Most of them are friendly, these green guys, but occasionally a bad one will pop up, one that has an AMR gene. And then these guys have a propensity to travel, so they'll creep across the skin, up into the urethra, cause a urinary tract infection. And some of these ascend even further into the donor kidney and cause a kidney infection and then even sepsis after that. So this is when things get really life-threatening. So we're going to do a study with patients who've received donor kidneys. And we're going to attempt to inspect the bacteria in the gut, in the bladder, and kidney and blood when they get infected to see what's happening. So how will the study work? Well, first, the patients will come in for their kidney transplant. The doctors, Reza and Mark, and their clinical team will take stool, urine, and blood samples before transplant, at transplant, and then 1 month, 3 months, 6 months, and a year afterwards, so we can track them in time. The biologists, Amber and Sanchuta, will work up the samples, extract the bacteria's DNA, and work out their genetic blueprints. The computational biologists, Lucy and Francois, will study the blueprints to see where the good bacteria are at any given snapshot in time, work out their signatures of AMR, and hopefully we'll find patterns in the bad ones and can work out how to recognize them. So what's the ultimate goal? Personalized medicine for kidney transplant patients, an early warning system. We can check the patient's stools early for the presence of the bad bugs, so the doctors can try to eliminate them with drugs before they start their migration up the bladder and beyond to cause all those problems. Our hope is that this will ultimately save donor kidneys and patients' lives.